So today we will learn the chapter 6. Chapter 6 title is The Diversity of Proteins. So in chapter 6 we will cover 4 subjects. The first one is prebiotic synthesis and origin of protein. Second one is evolutionary divergence of organism. And third one is protein sequence analysis. Finally, we will cover protein database. So in this slide, I will explain the first subject, prebiotic synthesis and the origin of the proteins. In early 1950s, Dr. Stanley Miller and Harold Urey performed experiment. So they just simulate prebiotic condition, especially the atmosphere condition in early Earth. So atmosphere in early Earth is composed of the methane and ammonia and hydrogen. And also the early atmosphere is composed of the water, H2O. Okay, so this early atmosphere is combined here. So finally they provoke the light spark, so that something is happen in this cylinder. Okay, so they cool down the reactions. Finally, water containing the synthesized biomolecule was precipitated. Okay, so after that, they analyzed what kind of biomolecule is synthesized in the water. So they found that this Table 6.1 summarized yields of biomolecules from simulating prebiotic conditions using a mixture of methane, ammonia, water, and hydrogen. Okay, several biomolecules were synthesized. Okay, among them you will see here. Okay, glycine was synthesized about the percentage about 2.1%. And also there is alanine is 1.7%, and also there was beta-alanine, okay, and also there is the glutamic acid, and also there is the aspartic acid, okay, what synthesized. Okay, so among the biomolecules there were 5 amino acids, especially the 4 amino acids, glycine, alanine, and glutamic acid and aspartic acid were synthesized during these reactions. So it means that the prebiotic synthesis of amino acids from simple organic molecules, especially the methane and ammonia and hydrogen and water. So after that we can synthesize the amino acid, especially the glycine and alanine and glutamic acid and aspartic acid. Okay, so this is the this subject is the summarize the prebiotic synthesis and the origin of proteins. So 4 amino acids were synthesized during this prebiotic synthesis. Okay. So in this slide, I will explain the second subject, evolutionary divergence of organism and its relationship to protein structure and function. So there are 2 different cells existed. Okay, one is prokaryotic cell, the other is eukaryotic cell. Okay, and prokaryotic cell is composed of bacteria and archaeobacteria. Okay, so what's the difference between the prokaryote and eukaryote? So big difference is that the first one is the membrane enclosed nucleus or membrane enclosed organelles. Okay, for example, the prokaryotic cells, so membrane enclosed nucleus is absent, and they are absent, and also membrane enclosed organelle is also absent. However, eukaryotic cells, So membrane enclosed nucleus is present, and the membrane enclosed organelle is also present. Okay, and also there are several differences between the prokaryote and eukaryote, but I will not cover in details. Okay, and also what's the big difference between bacteria and or archaeobacteria? What's the big difference? Okay, I will see the big difference in the next slide. Okay, the big difference between the bacteria and the archaeobacteria is the cell wall structure. For example, the bacteria, let me start the bacteria. So bacteria is composed of gram-positive and gram-negative bacteria. So this is gram-positive bacteria and this is gram-negative bacteria. Okay, for example, the gram-positive bacteria has the several peptidoglycan layers exist the outside the cytoplasmic membrane. So this is gram-positive bacteria, which has the cytoplasmic membrane, and at that outside the cytoplasmic membrane, there are several peptidoglycan layers exist the outside cytoplasmic membrane. How about the gram-negative bacteria? One or two peptidoglycan layers existed between the inner and outer membrane. Okay, that's why. So if we did crystal violet stainings, so crystal violet can stain the peptidoglycan layer, and also they can stain the space proteins, except that their color will be the crystal violet is like this. Okay, they will show that crystal violet color. How about the gram-negative bacteria? So crystal violet only can stain the space protein. That's why their color looks like the bright pink color is like this. So this is the gram-negative bacteria. How about the archaeobacteria? So for example, archaeobacteria, they have the pseudo peptidoglycan layer. The structure looks like the peptidoglycan, but is not exactly the peptidoglycan. So we call it as a pseudo peptidoglycan layer or the S layer. So S layer is composed of the proteins. So S layer exists the outside cytoplasmic membrane. Okay, let me summarize the difference between the bacteria versus archaeobacteria. The big difference is the cell wall structure. For example, bacteria has the peptidoglycan layer. However, archaeobacteria, they have the pseudopeptidoglycan layer or the S layer. Especially the bacteria, we can divide it into gram-positive bacteria and gram-negative bacteria. For example, gram-positive bacteria, they have the several peptidoglycan layers exist outside the cytoplasmic membrane, but gram-negative bacteria is one or two peptidoglycan layers exist between the inner and outer membrane. Okay. So in this slide, I will explain the third subject, protein sequence analysis. First, I will explain the protein sequencing. So protein sequencing, the common name, we call it as Edman degradations. So in the company, whenever we make the therapeutic antibody, so first we purify the protein. After that, we always do protein sequencing. So this protein sequencing procedure is very important. So you have to memorize all of the steps. Okay. So in this slide, I will cover the first and second step. In the next slide, I will cover third and fourth and fifth step. Okay. So first step is occurs in the mild alkaline condition. So pH is around 9. Okay, the reactant is PTC and N-residue peptide. The full name of PTC is the phenyl isothiocyanate. Okay, so PTC is react with N-residue peptides. So they will make the PTC derivative. So we call it as PTC 유도체. So PTC 유도체 is go to the second step. So second step is occurs in the acidic conditions. So for the second step, we change the buffer. So in the buffer, we add 3 fluoroacetic acids. So condition is changed to from the mild alkaline condition to the acidic condition. Okay, so in the acidic condition, PTC derivative is hydrolysis of PTC derivative happens. Okay, so for example, so this bond is attacked the carbon. After that, this bond is disconnected. So finally, they will make 2 yield. One is the labeled amino acid, and the other one will become the M-1 residue peptide. So this is the second step. So second step occurs in the acidic condition, especially we add the 3 fluoroacetic acid in the buffer. After that, hydrolysis of PTC derivatives happens. Hydrolysis happens, so this bond is disconnected. So finally, they will make the labeled amino acid and another M-1 residue peptide. So this is start from the R2, R3, R4. Finally, they have the Rn. Okay, so this is the second step. Okay. Okay, the third step in protein sequencing is also occurs in the acidic condition, but we additionally added 1 more HCl. So after we added 1 more HCl and the rearrangement of labeled amino acid happens. So okay, so this is the labeled amino acid. So okay, there is a sulfur and carbon. Between the sulfur and carbon, there is one bond, and this bond is disconnected. So, okay, this bond is disconnected, and the nitrogen has 1 pairs of electron, and this 1 pairs of electron is attacked this carbon. After that, rearrangement of labeled amino acid happens in the acidic conditions. So they will synthesize PTH amino acid. Okay, so you have to memorize the structure. It's okay. And fourth step is we just put this sample into the chromatography. After that, separation and elution of PTH amino acids is by chromatography. So maybe this peak contains this PTH amino acid. So after we separate the sample in the chromatography, we can elute this PTH amino acid. After that, we can identify this PTH amino acid. Okay, so we will know that what is R1. Okay, so this is the fourth step. Okay, during the fifth step, we can initiate another round of the protein sequencing by using the M-1 residue peptides. So N-1 residue peptides is they have the R2, R3, R4, finally they have the Rn. So this N-1 residue peptide is available for next cycle reaction with PTC. So okay, so they will initiate another round of the protein sequencing, which is composed of the first step and second step and third step and fourth step. Okay, so this is the full procedure of the protein sequencing, starting from the first step, second, third, fourth, and fifth. Okay. So in this slide, I will explain another protein sequencing method. So we call it as the mass spectrometry. So okay, mass spectrometry is composed of 7 steps. So in this slide, I will cover the first step. So first step is the enzyme digestion step. Okay, so protein extract can be digested with trypsin or other protease. Okay, but each protease enzyme has several characteristics. For example, trypsin. We usually use the trypsin in the mass spectrometry. Okay, trypsin is digest protein after arginine or after lysine residue. Okay, for example, here trypsin digest after arginine residue or after lysine residue. Okay, so this is figure 6.8 show that unknown protein. The timeline is digested with trypsin. Okay, so in this case we will see that this is an unknown protein. Okay, so after we treated with trypsins, or it digested after arginine, so R is arginine, and R is arginine, and R is arginine, and also if we treat trypsin, it can digest after lysine. Lysine is K, so after lysine, they can digest it like this. Okay. Okay. So after we treated the trypsins, so it will make the several peptide fragment. This fragment and this fragment and this fragment and this fragment is like this. Okay. And also secondly, we frequently used the second enzyme that we frequently used in the mass spectrometry is aspartate N. This is aspartate N. This enzyme is digested protein before aspartate residue. Okay, so this is aspartate N protein enzyme. So this is usually digested before aspartate. Okay, before aspartate. So in these cases, we will see that the aspartate is N is digested before aspartate. D is aspartate before aspartate, and before aspartate, before aspartate. So after we use aspartate N, so we can get another fragment is like this, which is composed of like this. Okay. So during the first step, we usually used trypsin. Okay. So trypsin digest protein after arginine or lysine residue. And also there are several other protein digestion enzyme, but we usually use the trypsin. Okay. Second one is the aspartate N, and there are also another enzyme is also available. So, but we will not cover in today's lecture. So during the second step, we can separate peptide fragments by liquid chromatography. So this is the one example of the liquid chromatography. Okay, so we can put mixture of peptide fragments in the liquid chromatography. After that, we can separate each peptide fragments by size. For example, in this case, we can elute the smaller fragment first, and also we can elute a medium fragment is next. Okay. So after that, we can separate. We can analyze the mass of each peptide fragment. So we can analyze each mass of each peptide fragment by mass spectrometry. So this is a mass spectrometry machine. So we can analyze the mass of each peptide fragment. So this is one example. For example, the smaller fragment is shown like this, and this medium fragment is shown like this. Okay, the x-axis is the mz value. Okay, so mz value is considered to be the mass of each peptide fragment. Okay, so because this fragment is smaller than this fragment, so they will shows here. Okay, and the y value is the relative intensity. For example, the smaller fragment has a large amount, so larger amount than the yellow fragment, the green fragment. That's why their peak is larger than this peak. Okay, so this is, we call it as experimental mass spectrometry analysis. So okay, during the second step and third step, we can separate peptide fragments by liquid chromatography. After that, we can analyze each mass of each peptide fragment like this. Okay. Okay. During the fourth step, we can theoretically digest protein sequences in protein database with trypsin or other proteases. So after we finished our human genome project, we get the protein database. So protein database is a collection of the protein sequences from all of the genes. So we have a protein database. So after that, we can theoretically digest the protein sequence in the protein database with trypsin or other proteases. So table 6.3 is one example. For example, we can theoretically digest unknown protein A with trypsin. After that, we can get this fragment, and this fragment, and this fragment, and this fragment, and this fragment is like this. So based on they have sequences like this. Based on this sequence, we can predict the mass of these fragments, and also we can predict mass of this fragment, and also we can predict mass of this fragment is like this. Step that we can do the fifth step. Fifth step is theoretical mass spectrometry analysis. Okay, so based on the prediction, so we can draw the theoretical mass spectrometry analysis. We can conduct theoretical mass spectrometry analysis. Okay, so this fragment will make this peak, and also this fragment will make this peak. Okay, and this fragment will make this peak. Maybe finally this fragment will make this peak is like this. Okay. So during the sixth step, we can compare the experimental mass spectrometry analysis with the theoretical mass spectrometry analysis. So we can compare A with B, A and B. Okay, so this is comparison. Okay, so this data comes from the experimental mass spectrometry analysis, and this data comes from the theoretical mass spectrometry analysis. So after that, we can find which peak is is matched each other, okay, we find that this peak is matched with each other. Okay, so based on this matched analysis, so maybe this peak we can predict that this peak comes from proteins A. Okay, so it means that this protein mixture contains some specific proteins A. So during the 7th step, we can identify some specific proteins based on this math comparison with the experimental mass spectral analysis and theoretical mass spectrometry analysis. Okay, so I explained full procedure of protein sequencing, especially the mass spectrometry, starting from step 1 to ended by step 7. Okay. So in this slide, I will explain how to identify proteins by using the experimental mass spectrometry data. So this is related to the problem number 4. So you have to understand this procedure to solve the problem number 4. Okay, so after mass spectrometry analysis, we get this experimental data. So, okay, so based on this data, we can predict that we have the one peptide fragment, which has the molecular mass is around 72 and 57 and 43 and 42 and 40 and 29 and 27 is like this. Okay, so now I will show you how to use this mass spectrometry data to identify proteins. Okay, and also second is we have to go to the Google and search the mascot peptide mass fingerprint. So this is the database. The people usually use this database to identify proteins by using the experimental mass spectrometry data. Okay. After that, you will go to this website. Okay. That third, you have to click this one, access mascot server, and also you have to click peptide, and also you will see that the peptide mass fingerprinter, and you have to click this one, perform the search. Okay, after that we have to choose the database. Okay, so there are several databases available, but we usually use Swiss plot database because this database contains most of the protein sequence. Next, you have to select the enzyme. Okay, so also when you use the trypsin, you have to click the trypsin. If you use the aspartate N, you have to select aspartate N. So it's okay. So you have to select the enzyme. So the other criteria, you just use the default setting. Don't change it. Okay. After that, you go to the query. Okay. Click the query. Okay. So in the experimental mass spectrometry data, we have this data. Okay, after that, we can input mass data here. Okay, just write down 72 or 57 and 30, 43 and 42, 41 and 29. After that, finally, you can click the start search. After that, you can identify proteins. Okay, so we can easily use this method to identify proteins by using the experimental mass spectrometry data. Okay, so in this slide, I will explain the alignment of protein sequences. So alignment of protein sequences allows us to measure similarities between protein and proteins. So this is the we already learned in chapter 3. So figure 3.2 show that primary sequence of the human myoglobin, and also they show that primary sequence of the whale myoglobins. So they compare A with B. So, okay, and then there will be the three cases. There is, for example, first one is identical residue, and there is also conservative transition, and also finally there is a non-conservative transition. So let me start identical residue. Okay, so if you see here the blue color, so it means that the sequence of the human myoglobin and the whale's myoglobin is exactly identical with each other. So in this case, we designated blue color is like this. Okay, how about the conservative transition? In conservative transition there are several cases, 5 cases. For example, first one is the amino acid containing the aliphatic side chains. For example, the isoleucine is changed to valine. So the amino acid containing aliphatic side chain, for example, isoleucine is changed to valine. In these cases, we call it as conservative transition. Another case is also, for example, here, this is the serine is changed to threonine. So in this case, the amino acid containing hydroxyl residue, serine is changed to threonine. So in this case, we call it as conservative transition. Another case is that the glutamate is changed to aspartate is changed to the glutamate. So amino acid containing acidic residue or aspartate D is changed to E. In these cases, we call it as conservative transition. Another case is that the amino acid containing basic residue, for example, lysine is changed to arginine. So we call it, in this case, we also call it as conservative transition. And also finally, there is the amino acid containing aromatic residue, for example, phenylalanine, tyrosine, and tryptophan. So in this case, phenylalanine is changed to Y. So in this case, we call it as a conservative transition. So we designated yellow color. Final case is the non-conservative transition. So except for these cases, we call it as a non-conservative transition. For example, glycine is changed to valine. So it means that there is no conservative transition. So we designated the red color. So based on these values, so we just can calculate we can just measure the similarity. Okay. I will explain one example in the alignment of protein sequence. So pairwise similarity searches or query sequence using protein database. Okay, so I will explain the detailed information. Okay, in figure 6.15 to show that the sequence of the alpha and beta chain of hemoglobin, so they aligned alpha with the beta chains. It's like this. Okay. So query sequence comes from the alpha chain of hemoglobin. The sequence is VLSPADKTNVKAW. Okay, but we assume that we don't know where this query sequence comes from. We don't know. Okay. Okay. So we use this query sequence, and we will do the pairwise similarity searches using the database. So this method is automatically identifies similar sequence and also calculate the similarity. Automatically calculate similarity. Okay, so for pairwise similarity searches of query sequence using protein database, first you have to go to the Google and just search is BLAST P. After that you will have this result. The protein BLAST and searches protein database using a protein query. So you can click this website. After that you will go to this website. You will show that a BLAST P should. Okay. After that, second is you have to enter query sequence here in these windows. Okay, so it's V, L, S, P. Finally, there is a K, A, A, W. So after that, you just can click the BLAST. After that, you will have. So after that, you will have this result page. Okay, so this is a query, and they search it. They used the protein database. After that, the result they show is like this. So this query sequence is the 100% identical with hemoglobin A2 comes from the Homo science, and also this query sequence is also 100% identical to transcription initiation factor IF2. And also you will see that there are several information is like this. Okay, so we can use the query sequence and we can do the pairwise similarity searches, and so we find that all of the result is like this. Except that you can click the alignment. Okay, after you click the alignment tab, so if you click the alignment tab, so you will see the detailed information of the pairwise similarity searches. Okay, so let me see here. So the query sequence was VLSP and KAAW, and this query sequence is exactly identical to the sequence of the hemoglobin alpha 2 chains, which comes from the Homo sapiens. Okay, so initially this query sequence comes from the hemoglobin alpha chain. But we assume that we don't know where the query sequence comes from. Finally, we found that this query sequence is 100% identical to hemoglobin alpha 2 chains. Okay. So from this slide, I will explain the procedure to compare two protein sequences. If we want to solve problem number 3, you have to understand this procedure. Problem number 3, the question was to compare the protein sequence of human alpha-lactalbumin with the protein sequence of the human lysozyme C. Okay, so to do that, first you have to go to the Google and search human alpha-lactalbumin. So this is the Google search result. Okay, there are several results will be there. So among them, so I usually click the uniplot.org, the result. So in the uniplot.org, you will see that alpha-lactalbumin precursor. So after you click this one, you will see this web page to show that uniplot, and the protein name was alpha-lactalbumin, and the organism was Homo sapiens. Okay, so if you scroll down this web page, okay, you will have this sequence. Okay, so this is so after you scroll down the web page, you will find the protein sequence of the human alpha-lactalbumins. So after that, you can just copy all of these sequence. Okay, copy the sequence of the human alpha-lactalbumin and paste it in the notepad. So you don't have to delete the number. Okay, just paste all of the information in the notepad. After that, you also have to find the protein sequence of the human lysozyme C and paste it in another notepad. It's okay. Okay, so after that, you can go to the Google once again, and after that, searches blast P. So after in the Google search result, you will see that the protein blast, and this protein blast can search its protein database using a protein query. So click this one, and you will go to the blast P web pages like this. Okay, now I will show you some important thing. Okay, and you have to click this one. After you click this one, align two or more sequence. So you will have two input windows like this. Okay. After that, in this window, you can input the protein sequence of the human alpha-lactalbumin, and also in this window, you can input the protein sequence of the human lysozyme C. So finally, you can click the blast. After that, you will see the result. Okay, so this is the full procedure to compare two protein sequences. So you have to understand the full procedure to solve the problem number 3. Okay. So in this slide, I will explain the structural homology arising from sequence similarity. So I will compare the cytochrome C sequence of the horse and the tuna and the yeast, and the P. dendrifer is a gram-negative bacteria, and also R. rubrum is also gram-negative bacteria. All of 5 species has cytochrome C, and their sequence they are nearly similar to each other, especially in tuna and horse. So only 17 out of 104 residue are different. It means that the sequence similarity between the tuna and horse is very higher. Okay, how about the structural homology? Okay, in next slide I will show you how to compare the structural homology. Okay, first we have to go to the Google and search the horse cytochrome C. So after that, you will see there several the Google result. So I usually click the uniplot.org. So just click this website, and finally you will see this website. And this is the cytochrome C, and it comes from the horse. After that, you can scroll down the web page, and after that, you will see the structure of the cytochrome C, human cytochrome C. So after you scroll down the web page, you will find the protein structure of horse cytochrome C. So they have the 4 alpha helix, and they have hem group is like this. Okay, how about the tuna cytochrome C? Okay, so in the Google, you also can search tuna cytochrome C. After that, you can click the uniplot. So you will, in the Google search page, you will see that uniplot, this is the cytochrome C, comes from tuna. And after that, after you click this result, you will go to this website. uniplot, and you will see that cytochrome C comes from the tuna. Okay. After that, you can scroll down the web page. After that, you will see that protein structure of the. After you scroll down, you will find the protein structure of tuna cytochrome C. They also have composed of 4 alpha helix, and they have hem group. And their structure, protein structure, is nearly the protein structure of tuna cytochrome C is nearly similar to the protein structure of the host cytochrome C. So it means that structural homology is arising from sequence similarity. Okay. So in this slide, I will explain the fourth subject, protein database. Okay. So in previous slide, we see that protein structure. So in the uniplot web page, we can easily see all of the protein structures. Okay, so most of the protein structure comes from the protein prediction. Okay, most of the protein structure comes from based on we can predict the protein structure based on the Chu-Passman algorithm. So Chu-Passman algorithm to show that the propensity use the propensity for a given amino acid residue to be found in the helix, strand, and turn. So table 6.7 summarizes the propensity for given amino acid residue to be found in the helix and strand, beta strand, or turn. For example, each amino acid has P alpha value and P beta value and PT value. Okay, so how about the proline? Proline has the P alpha value is 0.34, is very low, and P beta is 0.31. How about PT? PT is the 1.334. So PT is very larger than P beta and P alpha. Okay, so let me, table 6.8a shows that the algorithm of Chu-Passman. So let me summarize very important thing. Okay, the first thing is the second step. Okay, scan through the peptide and identify the region where 4 out of 6 residue have P alpha more than 1.0. In this region will be the alpha helix. Okay, so one example is there is the 6 residue. First one is alanine, second one is alanine, third one is arginine, and fourth one is also arginine, and the fifth one is asparagine, and sixth one is aspartate. So in this case, among 6 amino acids, 4 has the P alpha value is 1.0. So in this case, this region can be the alpha helix region. So this is the one basic concept. Another basic concept is the sixth step. Okay. So identify the region of sequence where 3 out of 5 residue have a value of P beta. P beta has the more than 1. Maybe this residue will be the beta strand. So, okay, so people used these 2 basic mechanisms, and after that we can predict where this sequence can be the alpha helix or where this sequence can be beta strand. So this is the prediction, but people usually found that the prediction is nearly similar to the real structure, comes from the X-ray crystallography. Okay, so through X-ray crystallography, so we can find the exact structure, but we found that the predicted structure is nearly similar to the X-ray, nearly similar to the structure comes from the X-ray crystallographies. Okay, so all of the protein structural prediction is based on the Chu-Passman algorithm. Just use the propensity for a given amino acid residue to be found in helix, strand, and turn. Okay, so there is a very important step is this one and this one. Okay, so today we learned chapter 6, specially focused on 4 subjects. First one is prebiotic synthesis and the origin of protein, and second one was the evolutionary divergence of organism. And especially we nearly focused on the protein sequence analysis. Finally, we briefly learned protein database. Okay,